Hi, everyone. Welcome to Foresight's Biotech and Health Extension seminar series sponsored by 100 Plus Capital. I'm so delighted to have Sergiy Velychko here today with us. We've recently been put in touch by the wonderful Fiona, who's also, I think, on this call. And yeah, I couldn't really quite believe what I saw when she shared a few blurbs and papers and stuff that you were working on. And we had a brief call. And yeah, I'm pretty shell-shocked by what you're working on. It has very long-term implications, but you're also making progress really in the short run. And so I'm really excited for your talk today on unraveling the secret of mammalian genesis. So please take it away. I will be in the chat monitoring for questions, and we'll hope that we leave a bunch of time for Q&A. Thank you so much for joining.
Thank you, Alison. Great pleasure to give some now to the Foresight community. Yeah. So the question is, why can't we engineer ourselves? Why do we remain as a wild type species evolved to be just a little more intelligent than an average monkey? And we actually can engineer mammals, and this is how majority of mammals are engineered today. This is done through the stem cell editing. The stem cells, the embryonic stem cells, can be derived from the early embryo. This stage is called blastocyst, and this inner cell mass of the blastocyst will become the future body. But you can get this embryonic stem cells and grow them indefinitely in the dish, and then use different gene editing technologies to edit those cells. And the nice thing is, even if 1 in 1 million cells is edited, you could still select the correct clone, expand it, grow it again indefinitely, possibly go through another round of edit, and then create an animal. But this only works for the mice. So all of this stem cell editing is possible for different species, including humans. But this last step, where we can create an animal from stem cells, is a limitation today. The mouse is by far the most edited species on the planet. And arguably, the true reason why this is the case is not because the editing technologies work better in mice than other species. It's because we have stem cells that we can culture and then create complete animals out of them. So the reason behind this is naive versus prime pluripotency. So the naive cells represent pre-implantation embryos, so earliest stage in development, and prime pluripotency represent post-implantation stage. So the first naive cells were derived by Martin Evans, the father of stem cells. He got a Nobel Prize for this work from the mouse blastocyst in 1981, so more than 40 years ago. And already in 1981, he used these cells to create the first mice, so the first mammals out of cultured cells. He called it creature out of culture. And this enabled the knockout mouse technology. So this technology absolutely was key to revolutionizing biomedical research. Majority of people who study mammals work on mouse because of this reason. But the other species, including human, are maintained in culture in this prime state. This was a challenge, and it still is a challenge. Shinya Yamanaka discovered the iPS cell technology in 2006. He also got a Nobel Prize for this work in 2012. He is perhaps one of the most famous Nobel Prize winner alive today. The iPS cell technology is just an amazing breakthrough. He showed that just by overexpression of 4 transcription factors, OCT4, SOX2, KLF4, and CMYC, it was possible to reprogram adult cells, such as skin or blood, into induced pluripotent stem cells. And these induced pluripotent stem cells are very similar to the embryonic stem cells, which means they can give rise to any tissue or organ of the body. So Yamanaka reduced the question of complete developmental reset to understanding these 4 transcription factors. So 2 years after his Nobel Prize winner work, Yamanaka's lab published this Nature Biotech paper where they showed that you could actually replace each of the Yamanaka factors with other factors of the same family. For example, SOX2 could be replaced with SOX1 or SOX3, KLF4 could be replaced, MYC could be omitted or replaced, but OCT4 was the only factor that could not be replaced by other OCT factors. So this made the whole field think that OCT4 is the most irreplaceable of the Yamanaka factors. So I published this paper, my first paper on this topic, and on the cover of Cell Stem Cell. And we showed that the field was actually partially wrong. You could completely omit OCT4 from the Yamanaka cocktail and still produce mouse iPS cells. But the most exciting part was that this SKM iPS cells were 20 times better at making complete animals comparing to the original OSKM iPS cells. So we could actually create complete animals from cultured stem cells right away using so-called tetraploid complementation experiment. And in this experiment, we create tetraploid embryo, which is capable of giving rise to functional trophectoderm and which will become a functional placenta. But this tetraploid embryo fails to create an animal. So tetraploid means that there is a double set of chromosomes, so twice more than it should be. And it seemed to not bother the placenta. Placenta doesn't care about being tetraploid. So now you can complement such tetraploid embryo with your cultured iPS cells, and they will occupy the inner cell mass and create a so-called all iPS cell animal. So complete. So this mouse was made in the end from skin because iPS cells were derived from skin. So we have derived the best quality iPS cell lines that has ever been published. And we achieved the highest cloning efficiency in animal that has ever been recorded, with up to 82% of live births, which is very much comparable to the natural conception rate and survival of natural embryos in the mouse. So this work was recognized by the German Stem Cell Network, and it was named one of the top technical advances of 2019 by The Scientist. And Shinya Yamanaka himself commented for the article in The Scientist that if we achieve the same for adult human cells, this could be a huge advantage for iPS cell technology. And indeed, 4 years later, we have achieved the same for human, but also for other species such as monkeys, cow, and pig. And this is this recent paper on Super-SOX that was also published in Cell Stem Cell around New Year's time. So what we found is that OCT4 actually has a double function. When it binds to DNA alone, it induces proliferation and priming, so exit from this highly developmentally potent naive state. But the dimer between OCT4 and SOX2 is the absolute core driver of naive pluripotency across species. We could stabilize this dimer by engineering highly cooperative Super-SOX, and this could allow more efficient generation of iPS cells across different species. We tested it in 5 species. And we also found that the SK cocktail, so SOX2 plus KLF4, even better with Super-SOX, is the minimal subset of Yamanaka cocktail that allows conversion of prime cells into naive cells for different species, including human. So this kind of explains the previous paper, because what we're suggesting here is that SKM iPS cells were more naive compared to OSKM iPS cells, and that explains why the SKM iPS cells were so much better at making animals. And by the way, by making animals cannot just apply for generation of complete animals out of stem cells. It's just the hardest core proof of pluripotency. If you can make a complete living animal out of your stem cells, this means that your stem cells can make any functional organ of the body. This started when we discovered that certain structural elements in SOX17, which is a SOX factor that cannot induce pluripotency, but certain structural elements from SOX17 could enhance induction of pluripotency when you swap them into SOX2. And the most interesting of such swaps was the single point mutation, alanine to valine at position 61. So the SOX2 has alanine and SOX17 has valine at this position. And we found that if you introduce just this point mutation, you can now generate iPS cells from with OCT factors that are not OCT4, including, for example, OCT6, which Yamanaka himself showed is normally incapable of making iPS cells. And this was very interesting because we do not change the OCT factor. We only modify the SOX factor, and somehow we enable reprogramming with other OCT factors. So the position 61V is located at the surface of the SOX2 molecule, where it interacts with OCT4 but only when they dimerize on this specific DNA motif, which is called canonical SOX-OCT motif, which is known to control majority of pluripotency genes. And when we showed by using molecular dynamic simulations that if you swap alanine to valine, you can increase the hydrophobic interactions between OCT4 and SOX2. And we also showed biochemically that this single swap can dramatically increase the stability of SOX-OCT dimer on the SOX-OCT DNA. And we showed it not just for OCT4, but for other OCT factors. And therefore, we explain why the SOX2 AV enables reprogramming with other OCT factors by forcing other OCT factors to interact with SOX2 similarly to how OCT4 does it. So the most exciting data came again from the making animals. So this is again tetraploidic complementation assay with different iPS cell lines generated using TET-inducible OSKM or episomal traceless OSKM, or the same vectors, but with a single amino acid swap. So as many other labs published before, including our lab, it is known that TET-inducible OSKM produces iPS cell lines that are not of good quality. Half of the lines are completely incapable of supporting full-term development, so the mice are never even born if you try to produce them from these lines. But even those that are born tend to die very quickly. So it's very hard or nearly impossible to engineer to get adult mice generated from lentiviral TET-inducible OSKM. So now you replace a single amino acid, a single letter from alanine to valine in the same identical vector, and you get all of the lines capable of supporting full-term development, and majority of the lines can produce adult mice that are completely healthy and normal, and they could breed. So with episomal vectors, which are traceless, so they don't integrate into the genome, they're very transient. They only exist around 1 week and then disappear. You can see that you can generate better quality iPS cells with this method, perhaps because there is no background leaking of the TET-inducible promoter. And you could see that even with episomal vectors, if you introduce a single point mutation, you could enhance generation of adult animals by 5 times. And since we characterize the single point mutation very well, we know exactly what it does. It enhances the stability of SOX-OCT dimer. We could basically make a model and postulate that this dimerization between OCT and SOX is the key driver of naive reset, and it's the key for generation of stem cells capable of making complete animals. So now we also created others. So we created a library of SOX2, SOX17 chimeras, and this SOX217 or Super-SOX was the most potent reprogrammer. It contains SOX2 61V replacement, but also a few other fragments from SOX17. So it could enhance reprogramming of human cells by 50 times. You could see it using either retroviral system or RNA system. It could also allow reprogramming with just 2 factors, OCT4 and Super-SOX, which could be also useful. It specifically could enhance reprogramming of old patient cells, which sometimes is very difficult using wild-type factors. So this is the episomal integration-free method. And it allowed generation of iPS cells for 3 new species. So these are the first iPS cells that are integration-free and generated with the cheapest method, the episomal method, for Cynomolgus macaque, which is the most important monkey for biomedical research. And these are also the first bovine iPS cells that are transgene independent and integration-free, which could be very useful for cow engineering or for growing meat in the dish. So we characterize these cells in details in the paper if you're interested in looking more details of these cells. The most exciting discovery of the paper is not even the Super-SOX, but what Super-SOX demonstrated. So in this case, these are just cell lines, pluripotent stem cell lines that were derived from pre-implantation embryo, mouse embryo, or post-implantation mouse embryo, so primed human iPS cells, and the primed cells converted into naive cells. And these are complete cell lysates. So we can lyse the cells, mix them with a DNA probe that is, for example, nanopromoter locus, and observe the content of the dimer that exists in this cell line. So none of these lines were treated with Super-SOX. And you can see that both human and mouse primed lines have a lot less dimerization between SOX2 and OCT4. And when you convert the mouse primed line into naive, so capable of making animals, you can see the dimerization is increased. And also the SOX2 expression is increased. Also, we, for the first time, explain how the 2I media works. So 2I media was invented in 2009 by the lab of Austin Smith. This media allows maintenance of naive cells for mouse and also rat. And nobody really knows how it works. And why doesn't it work for other species, only for rodents? And I think we now understand how it works. I think it just enhances the SOX-OCT dimerization. So using these insights, we developed a method that could enable generation of naive cells using episomes, so integration-free method. In this case, the minimal cocktail is SOX2 and KLF4, or even better, Super-SOX and KLF4. We also included mCherry in this plasmid so we can track the expression of the episomes, and you can deliver these episomes into the primed human iPS cells that many labs culture routinely. And within 1 week or even less, we can see reactivation of human-specific pre-implantation markers, such as SUSD2 and KLF17, which are not expressed in post-implantation prime cells. And you can nicely see that mCherry disappears, which means that the episome disappeared, and we have integration-free cells. You can see that this method is so robust, it works even in primed media, such as Stemflex or mTeaser. So this is media independent, and you can also confirm by gene expression that the SK specifically eliminate the episomes. This is very interesting property of the naive cells, that they can eliminate different exogenous DNA, including the episomal vectors. You can also see on the FACS data that naive media by itself did not produce SD2 positive population, but with mCherry SK cocktail, you could nicely have the population of the cells that is mCherry negative, but SUSD2 positive. So SUSD2 is a natural surface marker that can be used for sorting the converted cells, and you could potentially sort the cells and maybe make humans. But yeah, the question is, can the Super-SOX converted cells make humans? We, of course, could not answer that question for the paper. That would be illegal. But what we could do is to test the cells in cross-species chimerism. So what we did is we converted the cells with Super-SOX and sorted the SUSD2 positive cells. We marked them with constitutive RFP expression, so the human cells are red. And we aggregated them with early mouse embryos. And you can see that the majority of the embryos got human cells integrated in their inner cell mass. And in some cases, the whole epiblast was replaced with human iPS cells. So we have a very unique situation here where we have future placenta that is mouse, future yolk sac also mouse, but the future body is human, which is pretty cool. So the same method works for the cow. We could increase the developmental potential of cow iPS cells by overexpressing the same plasmid that we showed to work on human. And we showed that it enables the teratoma formation. Teratoma is a kind of tumor that contains all kinds of different tissues. So it's like distorted embryo. And this is a very good test for pluripotency. And we know that even cow embryonic stem cells are of very bad quality. They've been called losers in the recent paper by Jun Wu. And so they lose interspecies cell competition to human, while human loses to mouse. But it seems like this winner-loser status is controlled by SOX-OCT dimer, and the Super-SOX KLF4 cocktail could convert losers into winners. And this is the last experiment that I want to show. This is very convincing experiment because, so I didn't mention this before, but even for the mouse, the pluripotency was not truly solved. Some mouse lines can make mice and some cannot make mice. Nobody understands the difference. Of course, everybody would use only high quality mice, high quality mouse lines to produce animals. But nobody could understand what it means to be of high or good quality. So here we take a female line of a non-permissive strain, Female lines are known to be worse than male lines in producing animals, and we treat them with the same episomal cocktail that we just showed to work on human and cow. And at day 5, we use them for tetraploid complementation assay to produce all ESL mice. And you can see that M-Cherry SK produced 8 times more living pups compared to the M-Cherry control. So this is the first method that can force an existing low-quality pluripotent stem cell line into producing more living animals. So this is the take-home message. We showed that the SOX-OCT dimer is the key to the complete developmental reset. We could enhance it by generation of highly cooperative Super-SOX. And the SK cocktail is the minimal cocktail that allows generation of naive cells across species. So the advantage of germline editing through the stem cell route is it's the only method that potentially could allow indefinite multiplex editing, so multiple rounds. Even if you have a low editing efficiency, it is not a problem. So we could engineer designer mice way before the CRISPRs were online. And the reason is because stem cells allow in selection and quality control in culture before you make animals. And there is also potential for higher birth and survival rates comparing to the somatic cell nuclear transfer and other technologies. The advantages of Super-SOX naive reset is it's the only method that works in multiple species across different medias. It could enhance generation of live animals, and it uses the same mechanism as natural development. And you can read this Cell and Science papers that came to the conclusion that SOX2 OCT4 dimer is the key driver of induction of naive pluripotency in early development, early embryo. So these are my acknowledgements. Most of the work was done in Max Planck Institute in Münster, in the department of Hans Schöler, who discovered OCT4. And these are all the people who contributed to the study. So now coming to the applications. So how could we use naive cells for different species? So the most kind of obvious applications, the lower hanging fruit, is enhancing derivation of different tissues in the dish from human iPS cells. And we showed the proof of principle in this recent preprint by Marek Smela from Church Lab, where we showed that you could enhance the derivation of oogonia-like cells, which are precursors of eggs for 3 different iPS cell lines, including the iPS cell derived from George Church himself. And the naive reset cells produced oogonia-like cells that are the least methylated, which is a good sign. There is a big field now of trying to rejuvenate the whole bodies or tissues using partial cellular reprogramming, which kind of started by Ocampo and Belmonte paper in Cell, where they showed that the Yamanaka factors could ameliorate some hallmarks of progeria mouse model. So Yamanaka factors is the closest thing that we have to the fountain of youth. This is the most robust method of how to rejuvenate the cells. iPS cells generation is the ultimate rejuvenation. And the iPS cell fate resemble that of the youngest cells that ever exist in our body. And we showed that Super-SOX could specifically enhance reprogramming of aged human cells, enable 2-factor reprogramming, which could be useful because you might want to shrink down the cocktail for in vivo applications. We showed that SOX2-AV mutant is the only known modification of Yamanaka factor that can increase fidelity of the reprogramming process. And we showed that SOX2-AV makes the lentiviral inducible OSCAM cassette more compatible with long-term survival of the animal, which hints that maybe increasing cooperativity between OCT4 and SOX2 makes the OSCAM cassette less toxic to adult tissues. And there is some hints from recent papers by Vera Gorbunova and Skip Horvath, which both came to conclusion that SOX2, OCT4, and their direct target, NANOG, are upstream regulators of maximum lifespan in mammals. So it could be possible that SOX2 OCT4 dimerization is also key to the rejuvenation. For me, the most exciting outcome of the finding is the possibility of engineering animals beyond the mouse model. And of course, the most exciting would be to generate the first ever primate derived completely from stem cells. not only because this would be a proof of concept for human applications, that would be a proof that we finally have human cells that are truly pluripotent, capable of generating healthy organs, but also for future human applications for reproductive medicine. But also engineering monkeys would be very useful for modeling human diseases, particularly the brain diseases, because mice are the worst in modeling neurodegeneration, for example, which they don't have. Other applications could be engineering livestock or food, drug, and organ production. Genus, the company called Genus, has engineered pigs that are resistant to virus. It's a specific, very detrimental virus. But we could also use multiplex stem cell editing to increase productivity and animal welfare for more sustainable food production or drug production even. And also Egenesis, the company who recently transplanted the first pig engineered kidney into human living donor. And the other application would be to engineer the next generation of porcine donor for xenotransplantation. So when it comes to xenotransplantation, interestingly, the whole field is now focused on the pig for good reasons. Pig organs have a very similar size to human, and we can already engineer pigs through somatic cell nuclear transfer technology. But pigs are actually very distantly related to humans. They belong to different superclade of placental mammals with primates. So if you consider all animals, if you consider the evolutionary tree, of course, chimps would be the best animals to host human organs, but this is wouldn't be ethical, and also they reproduce very slowly. So I think primates are off the table. The second most closest clade of animals to humans are rodents and rabbits. And the largest of all rodents are by far capybaras. So capybaras make the most obvious choice for hosting human-sized organs. And they also already farm for food in South America. And the nice thing about capybaras is that they're just huge guinea pigs. So they're very much related to guinea pigs. And guinea pigs have a history of being great animal for experiments. They have contributed to 23 Nobel Prizes, mostly because the very similar immune system and the physiology to human. And I came to this independently just by looking at evolutionary tree, but people have thought about it before. This is the paper from 1994 that already discusses xenotransplantation and the more closer proximities of rodents to humans comparing to the pigs. And they even did some experiments showing that human blood reacts less strongly to capybara red blood comparing to the pig red blood cells. So I'm hopeful that Super-SOX will enable derivation of stem cells from guinea pigs and capybaras and could push forward this other possibilities for xenotransplantation. But the naive stem cells can even allow us to grow human patient-specific organs within animals through so-called interspecies embryonic chimerism. So in this case, you can, for example, take a pig embryo. This is theoretically, this has not yet been achieved. And you could knock out the possibility of this pig to create, for example, a pancreas. And then you can inject these embryos with human naive cells, patient-specific naive cells, and these cells could overtake the niche, the empty niche, and produce potentially human pancreas inside of a pig. So the proof of principle has been shown mostly on the rat and mouse. So people have generated rat pancreas inside the mouse, rat thymus inside the mouse, forebrain, kidneys. And the reason why this has mostly been done to mouse and rats is because first, they're more accessible experimental models. And second, these are the only two species for which we have true naive cells that could make animals. But interspecies embryonic chimerism can enable also xenopregnancy and the germline derivation that, for example, could be used for to aid species conservation. You might have heard of the success story of cloning of black-footed ferrets, which is an endangered species of ferrets, from frozen tissue samples. But the hidden hero of this work was the domestic ferret that was used as a surrogate to basically gestate this endangered species. And it was also shown that you could derive rat sperm inside of a mouse, or the opposite, mouse sperm inside of the rat. So you could potentially use a endangered animal as a surrogate for xenopregnancy or for derivation of the eggs and sperm of more endangered species. For example, one could imagine using domestic cat to gestate a snow leopard, or using less endangered or non-endangered rhino species to gestate an endangered rhino species. Or even in more extreme scenario, we could create a horse capable of gestating a rhino. We can go as far as even try to gestate a dolphin inside of a cow. Most of the people don't realize how closely related cows and dolphins are. Cows are more related to dolphins than, for example, they're related to horses or pigs. And the placentation inside in these hoofed animals is very superficial. It's the most superficial of all placental mammals. It's called epithelial coil. So this could potentially allow actually to, for example, dolphinize cow uterus to enable it to gestate a dolphin, a rare species of dolphin. So when it comes to genetic engineering of animals, I'm the most excited about finding the possible edits that could enhance performance, health, cognition of animals. This could potentially in future apply to the whole human population, unlike most of the work that is done today on genetic disease editing, which affects a relatively smaller proportion of population. But I think that of all species, dogs makes the most sense as a model for finding potential enhancing alleles. Dogs is the second most trainable species on the planet after human. They're also second most domesticated species on the planet after human. And they're second to human species that most commonly has jobs, which is pretty cool. This is also very likable species, and they have very quick gestation time. So you can test different enhancement alleles. Also, dogs have a lot of genetic diseases, more than any other species, because of extensive inbreeding. So we could potentially correct those diseases and make healthier dogs. Dogs, similarly to humans, have very different talents. Even within the same breed, different dog individuals are more talented than others. And for example, this was demonstrated in South Korean airport when they cloned a very talented drug detecting dog. His name was Toppy, and 100% of Toppy's clones were as good and as trainable as the original. So we could potentially derive iPS cells from such talented dog individuals and then further enhance them to not just to create clones, but to create super clones. And we could potentially enhance longevity, cognition, trainability, language understanding, athleticism. And how can we do it? There are already multiple studies that showed this possibility. For example, human language gene, FOXP2, has been shown to increase mouse intelligence and increase the complexity of mouse vocalization. And this was confirmed by multiple independent groups. It's very much possible that human version of FOXP2 could also enhance language understanding in dogs. Another possibility is overexpressing Yamanaka cocktail, specifically in neocortex or hippocampus in the brain, which was recently done in this preprint, and they were capable of generating smarter mice using this method, which is pretty cool because this is a very unnatural allele that could never be evolved by itself. So this is DOX-inducible Yamanaka expression in the neocortex. But one day, similar technology could be used in humans. When it comes to engineering different animals, I think megafauna is particularly interesting. I think that in the long-term future, the evolution of life on Earth will lead to creation of mega-intelligent megafauna. Like many successful species before in evolutionary history became megafauna. I think even humans one day will become giants to carry even bigger and more complex brains. But much before that, we could potentially create Ganesha by biosynthetically uplifting elephant brain. So uplift in the science fiction is the process of granting the human level intelligence to species that didn't evolve intelligence naturally. And the proof of principle of uplift has been successful in mice, rats, and monkeys. For example, this paper was on the cover of Science, where they showed that a single human gene could increase the size and folding of marmoset neocortex. So normally marmoset neocortex is completely flat. So we know that intelligent brain is basically architecture multiplied by scale. The elephant brain is around 5 kilograms at the moment. Potentially, they could carry a much larger brain, maybe even up to 20 kilograms. The surface of neocortex is around 4 times that of human, but the neocortex has much fewer neurons compared to human. But the nice thing is that we already know multiple human genes responsible for the complex architecture of neocortex, which we could apply to the elephant. I consider this as a safer path towards superintelligence compared to AGI, because at least the living beings will have the common interest of maintaining the habitable planet. And the initial experiments could be done on the rock hyrax. It's a species that is very closely related to the elephant, but it's the size of rabbit. It has much faster gestation and sexual maturation time, and it's non-endangered. It's even considered a pest in some countries. So this makes hyrax a perfect lab model for the future elephant application. So before we ever touch endangered elephants, we could verify the edits on rock hyrax. Thank you for attention. I'm happy to take questions.
Thank you so much. That was wild. Okay, let's see. We already have a few questions up here and then a few questions in the chat. Maybe I'll start with the questions in the chat. One person already had to leave, but Kriyan, do you want to ask one of yours?
Thanks, Allison. Anyway, yeah, a question I have is a really basic question. The mice that you were describing, they end up being clones of their single parents. Is that correct?
Yes.
Okay. Do they live a normal length life?
Yes. So the ones that survive, they live normal lives. Yeah.
Excellent. Interesting. Because as we know earlier, cloning didn't get that right.
So this is a very different technology to cloning, right? So the traditional cloning through somatic cell nuclear transfer takes just the nucleus. So you take a nucleus, you transfer into the egg, so you only reprogram the nucleus. And in this case, you reprogram the whole cell. So you convert the whole cell into pluripotent cells capable of making. But I do believe that the same technology could potentially also enhance the somatic cell nuclear transfer technology.
Okay. Gosh, I have so many questions. Allison, do you want to let someone else go first? Because I have 2 or 3 more questions. You're muted.
Allison, you're muted.
Sorry. Yeah, we'll get back to you in a sec then. How about Michael? Do you want to ask a question?
I just commented that it seemed like we could probably measure humans, and obviously humans have in a sense been domesticated for a longer time. But it seemed a little bit strong to say that they're the most domesticated species, and I was wondering whether we're basing that on genetic markers from things like the Russian fox experiment.
So to be honest, I'm not an evolutionary biologist. I think there is a big overlap between trainability and domestication. So I don't consider like species like chickens and like potatoes. So I'm considering like intelligent species. And I think it's only humans and dogs that are like that we are needed for our intelligence. And the self-domestication made us more trainable. That's why I said that human is the most domesticated species on the planet, just from the observation. This was not based on data, so sorry if that might have been wrong.
Okay, cool. And okay, Darwin definitely, with domestication, thinks of it as primarily about indiscriminate mating. That's like the primary thing that makes something domesticated. It doesn't need to be in a safe environment to reproduce. It to Darwin, but whatever. Anyway, other people.
Micah, how about you? Go ahead.
Sure. I also have lots of questions, but I'll just pick one. Is there any way to stop the oct from binding by itself at all, so it can only bind as part of the SOX-oct dimer? And if so, what would happen? Do you think it would just get even better? Is the problem the oct binding elsewhere, or is it there's some beneficial aspect and needs some oct binding by itself?
OCT4 alone binding induces proliferation, so you do need some level of proliferation. Is it possible to completely prevent it? Maybe you could engineer OCT4 that is dysfunctional unless it's cooperating with SOX2. We did some of these experiments by mutating some parts of OCT4, for example, linker domain. And in that case, only this highly cooperative SOX2 could enable reprogramming with such mutants. So we did similar experiments, but I don't think that disabling OCT4 would make it even better. I think what we need is to even further enhance dimerization. I think the dimer is more crucial than absence of the monomer.
Awesome. How about we go back to Creon and then we can go back to you, Micah?
Okay, so I have two more. I guess I'll just choose for one that might be more in your wheelhouse.
It's okay. We still have 10 minutes. Feel free to ask both of them.
Okay, I'll ask both of them in succession. One is, how much do we know about inducing partial pluripotency instead of total pluripotency? So instead of going, how much do we know about the factors that cause some cell to go back to full embryo cell, full, starting point cell versus that go partially back to, say, just regenerate a certain kind of organ without regenerating the whole organism? Have we unraveled some of that yet?
It's a very intense field of study. A lot of companies and groups are working on this, trying to find something a cocktail that would never produce iPS cells, or find a way to stop the Yamanaka factors at certain point. So the fully mature iPS cells are never produced, because those could potentially cause teratomas if they're induced in the body.
Of course. And so along those lines, I think that wound healing must hold a key to that, because when you get a wound, you get partial regression to stem cells, and it just redifferentiates in the best case to heal the wound.
Yeah, and then there is also a study that shows overexpression of OSCAM could enhance wound healing. Yeah, so
great. Okay, next question, and this is my last one, and it's a bit maybe farther off topic, but nevertheless related. Correct me if I'm wrong, but when a female mammal, placental mammal, or anything really, is conceived in the natural way and born, it has within itself at birth, and even before birth, its full set of ova and follicles and things like this. And somehow, if I'm correct, those particular cells are protected against aging, if you will, or stay in their pristine state, so that when the female later in life as an adult creates its own offspring, those offspring are young. So how is this protection? How is it that the egg cells in the female just don't age? Are they sequestered in some special environment? Do they have special factors that are expressed all during life or right before pregnancy or sexual maturity? What's going on?
And so this is a question about immortality of germline. It's not so both male and female germline, it's aging much slower than the rest of the body. It's not exactly true that they don't age at all. As you probably know, the older females produce a much less viable eggs. And this is also still a very active field of study. So I think that it's possible that the same mechanism is involved, so the same factors, because there is a big overlap between the germline and pluripotency. For example, OCT4 is expressed in both. Also, SOX factors and other factors basically expressed in both germline and pluripotent cells. It has even been shown that possibly the mouse germline at least can readily be reprogrammed with no factors, just like by culture conditions, into the pluripotent fate. Yeah, I think both fates are related, and it's possible that there is a overlapping set of factors that can keep the cells at the younger state.
Thank you. Great presentation.
Thank you.
Cool. Micah, it's you again.
At the moment, generating transgenic mice is very expensive and slow. How far away do you think the kind of discoveries you've made and your technique and whatnot are from replacing transgenic mouse generation in industry, specifically for mice that have a lot of edits? I've personally tried to get laboratories to give me mice with lots of edits, and the price just skyrockets the more edits you want. And so I'm very excited to hear anything you can say about where the future is going, specifically on transgenic mouse generation.
So this technology is, I think, most valuable because it will likely allow engineering other species similarly to how we do mice. But also we showed that it could potentially enhance the generation of mice, particularly through tetraploid complementation assay, which which could make it much faster, right? And there is at least one company that I know of in China, run by Guangming Wu, who generates mice immediately. So you save a lot of time because you immediately generate complete mice using the deployed complementation assay. So you don't need to wait for a few months for them to breed. So this is already possible in mice. But you can imagine that engineering other species could be as valuable or even more valuable as mice. And this work that shows how mouse lines are so developmentally potent can enable derivation of mouse-like embryonic stem cells for other species, including human, that could enable similar level of engineering.
Thanks. My other questions, I think, overlap with Allison, so I'll let Allison ask them.
Go for it, Micah. We still have 5 minutes. If you do, and if anyone else wants to raise hand, you're welcome to. I'll keep mine in the last minute or so.
So this is, Allison usually asks this, but I'll go ahead and ask it. Where can we throw money to make this research go faster and better? I'm a big fan of basically everything you listed as the kind of the future where this is going. How can people contribute or fund or just help out in any way?
Yeah, thanks. You could email me directly and we could discuss this. Yeah, I'm planning to deliver this to real world as soon as possible. I am considering founding a company as the most promising path, because in academia, it's almost inconceivable to work with animals that are not mice. Majority of academic labs are stuck with mice because it's very expensive to work with other animals. But there are some other entities, for example, primate centers. If you could fund primate centers to have more monkeys and to enable monkey experiments, that would be great.
Could you share a bit more your company? What would that focus on? Do you have any ideas or plans that you want to share?
Yes, so I'm trying to develop this as a platform, stem cell platform, which will enable all these different applications, not starting with Ganesha, of course. This is future possibility. But yeah, first we could just produce human stem cells that are better quality and also faster and more efficiently. Then we could also start engineering other animals. I would have monkeys as the priority, because this just means human applications that are the closest to human. So derivation of the first ever primates from stem cells would be a big milestone I will try to achieve. And this could potentially open new possibilities, such as improving the viability of human embryos. So using this for potential enhancing the human reproductive medicine. As you probably know right now, majority of the human embryos, the IVF embryos, are dying. 70% are dying. And for older females, it's 90%. So if we could increase the viability of the embryos, this would be huge for the field. But yeah, also I have potential collaborations with food industry that want to just create virus-resistant or more productive animals, and with xenotransplantation industry that wants to create next-generation xenotransplantation donors. So there will be multiple applications.
Yeah, I guess as a platform technology, it's like almost pretty unlimited there. Thank you so much. Is there anything that you wish we had asked that we didn't get to, or any way where people can reach out? I know that you already mentioned your email, but what's the best way for people to get in touch with you?
The best way is my email address, which is listed in the Super-SOX paper. Yeah, feel free to reach out.
Awesome. Thank you. And as usual, if you can't reach Sergiy, and if you're in this seminar room right now, feel free to email me, then I can try to make an introduction for you. Thank you so much. This was absolutely wild. And yeah, I wish you the best of success. I'm really excited for where this is going. And yeah, I'm excited for co-evolving a bit faster with other animal species and possibly much closer in the future. Thank you so much. I really appreciate it. Thanks everyone for your great questions, and I'll see you all soon.